Thank you, and I'd like to thank Matt and the rest of the organizing committee for inviting me here, and I don't see her here, but especially Lark for putting up with me when I changed my travel plans. Well, I was asked to give an overview of inflammation and then talk a little bit about my work. So we have, what, 2 days, Matt, to do that? So this talk might have been better placed at the beginning because we've already heard a lot about LPS and TNF, but we'll give this a try anyway. So I'm going to try to do this with a pointer here. So this is a slide that appeared in a review article a number of years ago, and it just encapsulates how we have thought about inflammation over the years as heat, redness, swelling, pain, and loss of function. And we still think of it that way in the broadest sense, but I think now it's become a lot more complicated, and we think of it more this way, and this is still a summary slide that is way oversimplified. But we think of inflammation now, and I have no idea where my pointer went, there it is, as arising from some sort of inflammatory stimulus. So we have a pathogen-associated molecular pattern molecules that are released from, as the name suggests, pathogens, microbes, oops. And we also have these damage-associated molecular pattern molecules, which you've heard a little bit about, that are released from typically from dying cells or from cells that have been injured. And these, LPS is one example of that, of course, released from gram-negative bacteria. And all these, we now think act at some sort of receptors. Toll-like receptors are the major category probably, but there are other receptors. And these receptors occur on a wide variety of cells. And when these cells are activated by these PAMPs and DAMPs, seem to be having a lot of trouble here. Okay. Leads to expression, well, activation of the cells and expression of a whole gaggle of inflammatory mediators and immune mediators. So we've got cytokines, various proteases, lipid mediators, reactive oxygen and nitrogen species, coagulation factors, which are going to be discussed later on, complement, you just heard about a little bit, and to quote the king and I, et cetera, et cetera, et cetera. So there's a lot more than I have up here, really. And one important thing to point out is that there are feedback mechanisms. So these mediators can feedback and further stimulate inflammatory or immune cells to release more of these agents, or they can also release agents that will turn off the inflammatory response, agents like interleukin-10 and prostaglandin E2, for example. But all this can culminate in what some of us like to call inflammatory stress, and if that is marked, it can cause tissue injury by itself. And this is injury not only to the liver, but you're all familiar with sepsis and multiple organ failure. We're going to get to you, just hang on. But if the inflammatory stress is modest, then it can be harmless or, of course, even beneficial if it leads to the elimination of pathogens. So that's my, my summary of the inflammatory response. And so the question here is, how can an innate immune response contribute to a toxic response? And this is really what I've been asked to talk about. And so starting with a xenobiotic agent, there are really a number of different ways this can happen. One is that an agent like LPS, for example, can actually bind to toll-like receptors, cause inflammation that is pronounced enough to cause injury to the liver. Another one is that a toxic dose can be, of a compound can be essentially an initiating event that may lead to altered homeostasis of the liver, maybe even some modest liver injury that will lead to the further injury by the release of these DAMPs from dying cells, causing inflammation and then further causing the progression of liver injury. Another way this can happen is from a non-toxic dose of a xenobiotic agent that can interact with an externally caused inflammatory response. And this can be due to again, the microbiome releasing LPS or even bacteria into the circulation, or some other external stimulus that is harmless by itself, interacting with a xenobiotic agent, which at a dose that's harmless by itself to cause liver injury from the combination. And then we have these inflammatory cytokines controlling xenobiotic metabolism. So many of you are familiar with the fact that cytochromes P450, for example, can be regulated by cytokines released during an inflammatory response. And of course, now in the past decade or so, transporters have become a major focus of interest, and these inflammatory cytokines similarly can control the expression of transporters. Okay, well, let's take some examples of these, and I'm not going to dwell on this one because we've already heard about LPS, and we know that LPS in high doses in animals and in people can cause liver injury through inciting an inflammatory response. An example of the next one would be acetaminophen, and we all think of acetaminophen as being metabolized to a toxic quinoneamine, which is responsible for hepatocellular injury, but recent studies have revealed that this can lead to the release of DAMPs from cells that are being injured, activation of toll-like receptors, and then the amplification of injury. So one DAMP is actually DNA chunks that can be released from dying cells, and they can activate toll-like receptor 9 and amplify liver injury. And here's just an example from the literature of a study by a Japanese group in which in wild-type animals we get liver injury, but in animals that are deficient in TLR9, that injury is much, much less. This is in the acetaminophen area, I think, an interesting but still controversial area of investigation. Okay, controlling cytochromes P450, and here's an example of that. These are some mice that were infected with a bacterium, and you can see in the I guess the stippled bars that most of these isoforms of cytochrome P450 were decreased in expression by this infection. That was not universally true, so a couple of them were not affected. One of them actually increased, so that I think the summary is that the predominant effect of these inflammatory cytokines on controlling CYPs is a downregulation, but there are some exceptions to that. And these effects of infection, as it says here, can be mimicked by inflammatory cytokines. And I'm not going to have time to show anything about transporters, but Eddie Morgan has written a nice review article, and the effects on transporters are similar. So the predominant influence is a downregulation of expression of transporters, but this is kind of a rule of thumb, and you have to look at individual transporters. So the idea here then is that an inflammation or infection can lead to a number of pro-inflammatory cytokines that can decrease P450s and decrease drug clearance. So we have cases where then an infection or inflammation, if you will decrease the clearance of a drug and leading to an increase in plasma concentration, getting above the threshold for toxicity and causing a toxic response. And there has been some reports in the literature of this happening in people. So there were cases, for example, back in I think about 1980 in Washington State, where kids who got the flu and were asthmatic and were taking theophylline for it actually experienced theophylline toxicity when the flu came on, presumably because of a decrease in theophylline clearance. Okay, well, I'm going to spend a bit more time on this one. And again, we're talking here about a non-injurious inflammatory response interacting with a non-toxic dose of a xenobiotic agent. And the idea here is that we again have release of inflammatory cytokines and other inflammatory mediators, but now instead of affecting the kinetics of the drug, one could view it as affecting the threshold for toxicity. So here we're not necessarily affecting kinetics, but if these inflammatory mediators are effective in decreasing the threshold for toxicity, then liver toxicity is going to ensue. And there have been a number of animal models now that we've developed and other folks have developed, most of which involve exposure to LPS as an inflammagen. So the idea is giving LPS at a non-hepatotoxic dose and coupling it with a non-hepatotoxic dose of a xenobiotic agent. And this table just represents, and I probably should have put the references here, but these are all studies that have shown interaction of inflammatory stress in these models with various agents. And as you look down here, you can see that plant toxins, industrial chemicals, various drugs, including some that cause idiosyncratic hepatotoxicity, some metals, and everybody's favorite environmental contaminant, dioxin, all interact with an inflammatory response to enhance toxicity. So I have just a few examples of that. Dave Eaton already mentioned aflatoxin, B1. Aflatoxins, of course, are known as hepatocarcinogens, but they also cause acute liver injury at high enough doses. And this was a study that Jim Lyondike did way back when he was wet behind the ears as a graduate student. Where are you, Jim? He's now in mid-career, but still wet behind the ears. But he looked at aflatoxin toxicity in rats, and what you can see here is, you get obviously a dose-dependent increase in ALT as a biomarker of liver injury. In the rats that were treated only with LPS, we see no liver injury, but what you see here is there's a marked shift in the left of the dose-response curve, so that doses of aflatoxin that were not hepatotoxic in the absence of inflammation become hepatotoxic in the face of a concurrent inflammatory episode. Acetaminophen, a similar thing, so we see a leftward shift. I have no idea where my pointer is. Here it is. A leftward shift in the dose-response curve. This is work that Jane Maddox did in the lab a number of years ago. And I might point out here that one thing about these models is that the timing of the inflammatory stress relative to the drug or xenobiotic exposure is absolutely critical. So in this study, we gave the LPS 2 hours before acetaminophen and saw this increase in toxicity. Others had reported previously that if you give endotoxin, it's actually protective, but they were giving it 24 or 48 hours before the administration of the drug. And this really makes sense if you think again about the inflammatory response being one where you get pro-inflammatory mediators released initially, and then those are all shut off, and you get an anti-inflammatory response to all that. Monocrotaline is a plant toxin. We've done a lot of work with that in my earlier career, and again, you can see here that in rats given monocrotaline, alone, you start seeing toxicity, also death out here, but when endotoxin is given along with it, you get a marked leftward shift in the dose-response curve. And then finally, Aaron Fullerton, working in Patty Gainey's lab, worked with a different model of inflammation, or really it's more related to adaptive immunity, or at least lymphocytes. Concanavalin A produces a little bit of toxicity by itself at the dose that Aaron used. The TCDD, which he was interested in, produced no toxicity at the doses. So this is one dose of TCDD given 4 days before the concanavalin A challenge. But you can see that fairly small doses of TCDD were able to enhance the concanavalin A hepatotoxicity. So I guess the point here is that there are a number of really unrelated agents, hepatotoxicants, the toxicity of which can be markedly enhanced during inflammatory stress. So I'd like to talk a little bit about Diclofenac. So we've become interested in the last few years in how this interaction with inflammatory stress may underlie some idiosyncratic drug-induced liver injury reactions. And Diclofenac, as it says here, is a nonsteroidal anti-inflammatory drug that's caused idiosyncratic drug-induced liver injury in human patients. And interestingly, I think, osteoarthritis has been identified as a susceptibility factor. And of course, this is a condition that's involved with inflammatory responses. Now, in vitro, large concentrations of Diclofenac can cause cell death, and this is mediated by activation of apoptotic pathways and mitochondrial permeability transition that ends up killing cells. And unlike a lot of other drugs that cause idiosyncratic hepatotoxicity, if you give this drug to rats at high enough doses, it actually will cause liver injury. And this is a study that we did a number of years ago, and I've lost my pointer again. And what you can see here, this is again in rats given 100 milligrams per kilogram of Diclofenac, you get an increase in serum ALT that's accompanied by histologic evidence of hepatocellular necrosis. And what we found was you could actually culture bacteria out of these livers. I don't have the controls on here, but we were able to culture considerable numbers of bacteria from the livers of Diclofenac-treated rats. And when we gave non-absorbable antibiotics, that decreased the bacteria we're able to culture, and it also markedly reduced the liver injury. So you see in the antibiotic-treated animals, they experience less liver injury. So we then turned and asked if we gave a smaller non-toxic dose of Diclofenac, if we could render those animals susceptible by a concurrent administration of LPS. And here what you see is that that's what we found. So these are animals given either the LPS alone or the Diclofenac alone. At this dose, no toxicity occurred, but in the combination, we got an increase in serum ALT and, again, histologic evidence of hepatocellular injury. And work from Angela Mali's group in Germany actually confirmed this even at lower doses of Diclofenac. So she gave Diclofenac at 7.5 milligrams per kilogram per day for 7 days and essentially found the same sort of increase in toxicity upon exposure to LPS. And also in that publication, she looked at a number of inflammatory mediators and found that in the co-treated animals that there were greater expression of a number of these pro-inflammatory mediators. And this was of interest to us, especially these last two, TNF and interferon-gamma, because we had shown with another drug in a mouse model with actually trovafloxacin that you get an increase in TNF, an increase in interferon-gamma, and elimination of these protected against the synergistic toxicity between trovafloxacin and LPS. So subsequently, some work from Lisa Fredriksen's group in the Netherlands really addressed one possibility out of many in terms of how this interaction works. And that is one way, but certainly not the only way, is that the drug can be increasing the susceptibility of hepatocytes to injury from these cytokines. So they examined TNF, and if I can find my pointer again, here you go. Okay, so if you look at the open bars, these are in HepG2 cells, and if you dump enough Diclofenac on these cells, you do get injury. But what they're showing here is that in the face of added TNF, which of course is one of the inflammatory mediators that seems to be important, given alone doesn't produce any cytotoxicity, but it enhances or renders cytotoxic some of the concentrations of Diclofenac that are normally not cytotoxic. So what's going on here? Well, what does TNF do, first of all? So TNF acts at a couple of receptors, and especially receptor 1 is thought to activate apoptotic pathways, which you see here, which can lead, of course, to cell death, and can also lead to the activation of JNK, which is not harmful by itself unless that activation is prolonged, in which case it can contribute to cell death. So why don't cells die when you dump TNF on them? Well, the reason is because it also activates anti-apoptotic pathways. So it activates NF-kappa B, which in turn causes the expression of target genes that basically inhibit the apoptotic response. And I'm not going to have time to show you the data, but what Frederickson's group showed was that the Diclofenac can actually inhibit this inhibitory pathway. And in addition, Diclofenac causes ER stress in cells, which can activate PERK and CHOP, which then leads to the expression of apoptotic factors. It also causes the prolonged activation of JNK, which can contribute to this process. Okay, so that brings us finally to some work that we're engaged with right now, and Ashley Maori is here somewhere over there. She'll be looking for a postdoc soon, so keep her in mind. So we were interested in what role interferon-gamma plays in all this, because again, we had shown in a couple of models that both TNF and interferon-gamma seem to be important. So this is a study that she's done in, again, HepG2 cells. And what you see here is that at these concentrations of Diclofenac, we don't get any cytotoxicity, but when they're exposed concurrently to a concentration of TNF that's non-toxic, we get a cytotoxic response, just like the Frederickson group showed. But what was interesting is that the combination of interferon-gamma and the drug didn't do anything. So the interferon-gamma was not effective by itself in enhancing toxicity, but what it did do is enhance the cytotoxic effect of the TNF drug combination. And we've been interested in, these are really preliminary data, none of this is published yet, but we were able to confirm in these cells the importance of JNK in the response to TNF. Okay, so these are cells that were treated with either Diclofenac alone or in combination with TNF, and you see the drug TNF combination led to cytotoxicity, which was inhibited by a JNK inhibitor. And interestingly, the combination with interferon-gamma, when interferon-gamma was in the picture, that the JNK also seems to be important because it markedly reduced the toxicity. What was a little more surprising was studies with ERK. So Diclofenac also seems to cause a prolonged activation of ERK. So this is 12 hours and 18 hours phospho-ERK is increased by the drug alone, not affected perhaps too much by the cytokines. Remember, the drug alone doesn't cause any toxicity. And we normally think of ERK as a cytoprotective, being cytoprotective, but that's not always the case. And here's a good example of that. Again, my pointer is, here we go. So surprisingly, an ERK inhibitor, it's really a MEK inhibitor that inhibits the phosphorylation of ERK, was ineffective in influencing the TNF drug interaction, but it did markedly decrease, I'm really having trouble with this, okay, markedly decrease the interaction when interferon-gamma was on board. So it looks like ERK is playing a cytotoxic role here, if you will, when interferon-gamma and TNF are interacting with the drug. And Ashley's gone on to look at a number of other agents. So a caspase inhibitor also reduces this response. If you chelate intracellular calcium, it reduces the response. I'm sorry, that's a caspase inhibitor. The calpain inhibitor is down here. Interestingly, inhibitors of nitric oxide synthase or an SOD mimetic did not influence the toxicity, at least not in our hands yet. And again, please take these as somewhat preliminary, but they seem to keep holding. So what our preliminary studies then have shown is that we get an activation of calcium, activation of calpain, which is probably in this ER stress pathway that was identified by Lisa Fredrickson and her group. And we also have an activation of ERK. And again, ERK usually, when it's phosphorylated, will translocate to the nucleus and activate genes that are involved in cytoprotective pathways. But here it doesn't seem to be doing that. So one of the questions is, and we haven't answered this question by any stretch of the imagination, so please note the working hypothesis up at the top. We have no data to support what I'm about to show you, but this is kind of where we're going with this anyway. So what happens when interferon-gamma is on board? Well, interferon-gamma can activate pathways that will prevent the translocation of phosphorylated ERK into the nucleus. And this is not our work, it's work that has appeared in the literature. And so if interferon-gamma is doing this, it may lead to a buildup of phosphorylated ERK in the cytoplasm, and we know that when that happens, that can activate other kinases. That is a death-associated protein kinase that is can be responsible or at least contribute to apoptosis. So this is one way, after scratching our heads a lot, that we can explain these rather strange data. So just to wrap up then, interferon-gamma and other toxic and inflammatory stress interaction models. I mentioned the trovafloxacin model in which we co-treat mice with LPS, and we get an enhancement of TNF and interferon-gamma. Let me put this a different way. When we treat with LPS, we get an increase in TNF and interferon-gamma, and that increase is actually prolonged or enhanced by trovafloxacin. And as I mentioned, elimination of either TNF or interferon-gamma eliminated the liver injury. And then in vitro, we've shown recently that trovafloxacin actually causes DNA damage and activates ATR, another kinase, and in vitro it synergizes with TNF to cause cytotoxicity. And Ashley's shown that in combination with TNF, it led to prolonged activation of MAP kinases, and that the cytotoxic interaction was enhanced by interferon-gamma. In a model of halothane hepatotoxicity in mice, and this is some of Patty Gainey's work, Christine Dugan in her lab showed that liver injury from halothane depended on interferon-gamma and NK cells. And then another group has published interaction of LPS with doxorubicin, which leads to hepatotoxicity in mice and is related to enhanced interferon-gamma production and neutralization of interferon-gamma afforded protection. And then finally, I showed you some results with TCDD and Con-A as the inflammatory stimulus, if you will. Gisela Teagues has spent a career on Con-A, showing that the toxicity from high doses of that are dependent on interferon-gamma and TNF, and inflammatory cells like macrophages and NK T cells, but also CD4 positive T cells. And what Aaron Fullerton showed is that TCDD treatment, pre-treatment, as I've already shown you, enhanced toxicity, but it also enhanced interferon-gamma production in vivo, and it also invoked NK cell involvement. So NK cells are one cell type that can produce interferon-gamma, and it turned out that depletion of NK cells was protective. So in summary, inflammation can contribute to hepatotoxicity in a number of different ways. An episode of inflammation can sensitize the liver to injury from various toxic chemicals. And as I mentioned, the timing and the magnitude of the inflammatory episode are really important in whether or not you get a toxic outcome. Hepatocytes stressed by a toxin become sensitized to death from inflammatory mediators, and TNF is a critical mediator, and interferon-gamma seems to enhance that killing ability, at least with some compounds. And acting together, these toxicants and inflammatory mediators can activate cell death signaling pathways. And I'll stop there. I'd like to acknowledge my faculty colleagues, Patty, who's here, and also Jane Maddox, who did the acetaminophen work, and again, Jim, Steve Yee, Sean Dang, Pat Shaw, Aaron, whom I mentioned, and Ashley, and a couple of undergraduates, actually, from Germany, who were visiting MSU, contributed to this work, and of course, support from NIH. Now, thank you, and I'll be glad to take any questions. Dave?
So, obviously, a lot of work has been done in hepatotoxicity and in vitro cell systems that are purified hepatocytes. Can you mimic some of the inflammatory roles by cultures with cells?
We've done some of that, as have others, and the answer to that is yes, with at least some of these compounds. So with trovafloxacin, for example, when we co-cultured Kupffer cells, and I think these were rat hepatocytes, with trovafloxacin, and then stimulated the Kupffer cells with LPS, they produced TNF, and we got cytotoxicity in the hepatocytes. So I think we did this originally in rat hepatocytes, and then more recently we've used these spheroids that are marketed now. I know that.
Bob, in this model of the LPS with hepatotoxin, I have a two-part question. Is there anything unusual about the histopathology of what happens in the liver?
Is there anything unusual?
Yes. So let me give you the second part.
Okay.
So it's very interesting with the acetaminophen. I've been a part of several studies of chronic hepatotoxicity with acetaminophen. And, for example, a one-year study in mice where the animals had profound central lobular necrosis, virtually no inflammation. And the same is true in humans. I've seen cases of individuals who repeatedly tried to commit suicide with acetaminophen, and when they finally succeed after a few tries, there's no liver inflammation. So I'm wondering specifically with the model you did with LPS and acetaminophen, was there then inflammation with the addition of LPS?
Yeah, that's an easy question with a complicated answer. But the answer is yes, we saw inflammation in those livers of animals that were co-treated with LPS and acetaminophen. And the inflammation was predominantly neutrophilic. We actually don't know what the role of the neutrophils is in that model. So I don't think we ever did neutrophil depletion, did we, in that model? No, acetaminophen. Yeah, so, and the role of neutrophils, especially in acetaminophen toxicity, is very controversial. But I think the prevailing opinion at the moment is that they're probably there to clean up, at least with acetaminophen alone at high doses. The situation might be much different when you have inflammation interacting with a non-toxic dose, usually non-toxic dose of acetaminophen. But your first question, is there anything unusual? Well, the inflammation is, of course, greater when you have the LPS there, because that's going to bring in inflammatory cells. But in some of these other models, for example, in the aflatoxin LPS model, the lesion you see looks like high-dose aflatoxin. It's centered around the periportal area, maybe a few more inflammatory cells there, but it's still still an aflatoxin-like lesion. You get injury to bile ducts as well as hepatocytes. In the monocrotaline model, it's very different. It actually looks like a hybrid lesion between that you get with high-dose endotoxin or high doses of monocrotaline. So I need to get off the stage here, but LPS produces a mid-zonal lesion in high doses. Monocrotaline produces a central lobular lesion, and you see both of those arising when you give those two together. So even though lesions seem to be a little bit different depending on the toxicant and probably how they interact.
There are a lot of questions, and we could answer some of them at break. You want to flip up the next one here?